A colorimeter is an instrument that measures the amount of light absorbed by a colored solution. It works based on the Beer-Lambert law, which states that the amount of light absorbed by a solution is directly proportional to the concentration of the absorbing substance and the path length of the light through the solution. The main components of a colorimeter are: light source, this provides the light that is used to illuminate the sample solution. A tungsten filament lamp is commonly used as the light source in colorimeters. Monochromator, this is used to select a specific wavelength of light from the light source. This is important because the absorbance of a solution is wavelength dependent. Cuvette holder, this is where the sample solution is placed. Cuvettes are typically made of glass or plastic and are transparent to the wavelength of light that is being used. Photodetector, this detects the amount of light that is transmitted through the sample solution. The signal from the photodetector is then amplified and displayed by a meter or readout device. The Beer-Lambert law states that the absorbance of a solution is directly proportional to the concentration of the absorbing substance and the path length of the light through the solution. The Beer-Lambert law can be expressed mathematically as follows. A equals epsilon Bc, where A is the absorbance of the solution. Epsilon is the molar absorption coefficient, which is a property of the absorbing substance. B is the path length of the light through the solution, in centimeters. C is the concentration of the absorbing substance, in moles per liter. Question 2. Write about the components of lipid profile with normal reference range. Add a note on significance of LDL. Answer: normal lipid profile is total cholesterol, below 200 mg/dl. High density lipoprotein, HDL, cholesterol, above 60 mg/dl. Low density lipoprotein, LDL, cholesterol, below 100 mg/dl, for people who have diabetes, below 70 mg/dl. Triglycerides, below 150 mg/dl. LDL stands for low density lipoprotein. It is a type of lipoprotein that carries cholesterol throughout the body. LDL is sometimes called the bad cholesterol because high levels of LDL cholesterol can build up in the walls of your arteries. This buildup is called plaque, and it can cause health problems, such as heart disease and stroke. LDL cholesterol is made in the liver and is used by cells throughout the body to build cell membranes and make hormones. However, too much LDL cholesterol can build up in the blood and form plaque in the arteries. This can narrow the arteries and make it harder for blood to flow through them. Question 3. Classify carbohydrates. Add a note on lactose intolerance. Answer: the carbohydrates are further classified into simple and complex, which is mainly based on their chemical structure and degree of polymerization. Simple carbohydrates: 1. Monosaccharides, 2. Disaccharides, and 3. Oligosaccharides. Simple carbohydrates have 1 or 2 sugar molecules. In simple carbohydrates, molecules are digested and converted quickly, resulting in a rise in the blood sugar levels. They are abundantly found in milk products, beer, fruits, refined sugars, candies, etc. These carbohydrates are called empty calories, as they do not possess fiber, vitamins, and minerals. Complex carbohydrates, polysaccharides. Complex carbohydrates have 2 or more sugar molecules, hence they are referred to as starchy foods. In complex carbohydrates, molecules are digested and converted slowly compared to simple carbohydrates. They are abundantly found in lentils, beans, peanuts, potatoes, peas, corn, whole grain bread, cereals, etc. Polysaccharides are complex carbohydrates formed by the polymerization of a large number of monomers. Examples of polysaccharides include starch, glycogen, cellulose, etc. Lactose intolerance is a condition in which your body cannot digest lactose, a sugar found in milk and dairy products. This is because your small intestine does not produce enough of the enzyme lactase, which is needed to break down lactose. Symptoms of lactose intolerance. The symptoms of lactose intolerance can vary from person to person and may include bloating, gas, diarrhea, abdominal cramps, nausea, vomiting. Question 4. Classify amino acids with examples. Answer: there are 3 types of amino acids. Essential amino acids: these are the amino acids that cannot be synthesized by the body and must be obtained from the diet. The essential amino acids are histidine, isoleucine, leucine, lysine, methionine, phenylalanine. Threonine, tryptophan, valine. Semi-essential amino acids: these are the amino acids that can be synthesized by the body, but not in sufficient amounts. The semi-essential amino acids are arginine, cysteine. Non-essential amino acids: these are the amino acids that can be synthesized by the body in sufficient amounts. The non-essential amino acids are alanine, asparagine, aspartic acid, glutamic acid, glutamine. Glycine, proline, serine, tyrosine. Question 5. Explain in detail about the factors affecting enzyme activity. Answer: factors that affect enzyme activity are enzyme concentration: the more enzyme present, the faster the reaction will proceed. This is because there will be more enzyme molecules available to bind to the substrate and catalyze the reaction. Substrate concentration: the more substrate present, the faster the reaction will proceed, up to a point. This is because there will be more substrate molecules available for the enzyme to bind to. However, if there is too much substrate, the enzyme molecules will be saturated and the reaction rate will not increase any further. Temperature: enzymes work best at an optimal temperature. This temperature is specific for each enzyme and is usually around 37 degrees Celsius, 98.6 degrees Fahrenheit. If the temperature is too low, the enzyme molecules will not be able to move around and bind to the substrate. If the temperature is too high, the enzyme molecules will denature and lose their shape. pH: enzymes also work best at an optimal pH. This pH is specific for each enzyme and is usually around neutral, pH 7. If the pH is too acidic or too alkaline, the enzyme molecules will lose their shape and become inactive. Cofactors and coenzymes: some enzymes require cofactors or coenzymes to function properly. Cofactors are inorganic ions, such as metal ions, while coenzymes are organic molecules. If the cofactor or coenzyme is missing, the enzyme will not be able to function. Inhibitors: inhibitors are molecules that can bind to enzymes and slow down or stop their activity. There are 2 types of inhibitors: competitive inhibitors and non-competitive inhibitors. Competitive inhibitors bind to the active site of the enzyme, preventing the substrate from binding. Non-competitive inhibitors bind to a different site on the enzyme, changing its shape and making it less active. Question 6. Write about the sources, RDA, and deficiency disorders of vitamin D. Vitamin D sources include fish, mushrooms exposed to sunlight, fortified milk substitutes, fortified tofu, fortified yogurt, fortified breakfast cereals, fortified orange juice, pork chops, and eggs. The recommended dietary allowance, RDA, for vitamin D is 15 micrograms, mg, for adults and 10 mg for children under the age of 12. The RDA is the amount of a nutrient that is enough to meet the needs of most healthy people. Vitamin D deficiency is common worldwide. It is estimated that about 1 billion people worldwide have vitamin D deficiency. Vitamin D deficiency can lead to a number of health problems, including rickets. Rickets is a disease that causes the bones to become soft and weak. It is most common in children, but it can also occur in adults. Osteomalacia. Osteomalacia is a disease that causes the bones to become soft and weak. It is most common in adults, but it can also occur in children. Osteoporosis. Osteoporosis is a disease that causes the bones to become weak and brittle. It is most common in women after menopause, but it can also occur in men and women of all ages. Muscle weakness, vitamin D deficiency can also lead to muscle weakness. Fatigue, vitamin D deficiency can also lead to fatigue. Question 7. With the help of a neat labeled diagram, explain the types of weighing balances. Answer. Examples of laboratory balances include the triple beam balance, analytical balance, micro and semi-micro balances. Question 8. Write about phlebotomy, specimen collection, storage, transport, and disposal of needles. Answer. Phlebotomy is the process of drawing blood from a vein. It is a common medical procedure that is used to collect blood for testing, donation, or transfusion. Specimen collection is the process of obtaining a sample of tissue, fluid, or other material from a patient for diagnostic testing. The type of specimen collected will depend on the test that is being ordered. Storage of specimens is important to ensure that they remain viable and uncontaminated for testing. The specific storage requirements will vary depending on the type of specimen. Transport of specimens is also important to ensure that they remain viable and uncontaminated during transit. The specific transport requirements will vary depending on the type of specimen and the distance it is being transported. Disposal of needles is important to prevent needle stick injuries and the spread of infection. Needles should be disposed of in a sharps container immediately after use. Question 9. What are centrifuges? Mention the types and uses of centrifuges. Answer. A centrifuge is a laboratory instrument that uses centrifugal force to separate mixtures of liquids or solids. The centrifugal force is created by rotating the sample at high speed. The heavier particles in the mixture will move to the outside of the centrifuge tube, while the lighter particles will move to the inside. There are different types of centrifuges, each with its own specific uses. Some of the most common types of centrifuges include microcentrifuges. These are small centrifuges that are used to spin small samples, such as blood or urine. Microcentrifuge laboratory equipment. Benchtop centrifuges. These are larger centrifuges that are used to spin larger samples, such as blood plasma or cell cultures. Benchtop centrifuge laboratory equipment. High-speed centrifuges. These are very powerful centrifuges that are used to spin samples at very high speeds. They are used for applications such as DNA separation and protein purification. High-speed centrifuge laboratory equipment. Ultracentrifuges. These are the most powerful centrifuges. They are used to spin samples at extremely high speeds, up to 100,000 rpm. They are used for applications such as virus isolation and subcellular fractionation. Ultracentrifuge laboratory equipment. Centrifuges are used in a wide variety of applications, including medical diagnostics. Centrifuges are used to separate blood cells, plasma, and serum. They are also used to separate urine components. Biological research. Centrifuges are used to separate DNA, RNA, and proteins. They are also used to isolate cells and organelles. Industrial applications. Centrifuges are used to separate solids from liquids in a variety of industrial processes, such as the production of food, beverages, and pharmaceuticals. Environmental applications. Centrifuges are used to separate pollutants from water and wastewater. Question 10. What are nucleosides and nucleotides? Give examples. Nucleotides and nucleosides are the building blocks of nucleic acids, which are DNA and RNA. Nucleosides are composed of a nitrogenous base, a 5-carbon sugar, and a phosphate group. The nitrogenous bases are either purines or pyrimidines. Purines are double-ringed structures, while pyrimidines are single-ringed structures. The 5-carbon sugar is either ribose or deoxyribose. Ribose is found in RNA, while deoxyribose is found in DNA. Nucleotides are composed of a nucleoside plus one or more phosphate groups. The phosphate groups are attached to the sugar molecule at the 5-carbon. The number of phosphate groups determines the type of nucleotide. Mononucleotides have one phosphate group, dinucleotides have two phosphate groups, and so on. Examples of nucleosides and nucleotides. Nucleosides: adenosine, guanosine, cytidine, uridine, and thymidine. Nucleotides: adenosine monophosphate, AMP, guanosine monophosphate, GMP, cytidine monophosphate, CMP, uridine monophosphate, UMP, and thymidine monophosphate, TMP. Question 11. Write briefly about anticoagulants. Anticoagulants are medications that prevent blood clots from forming. They are used to treat and prevent a variety of conditions, including deep vein thrombosis, DVT, pulmonary embolism, PE, stroke, and heart attack. There are two main types of anticoagulants. Heparin. Heparin is a natural substance that is made by the body. It works by blocking the action of a protein called thrombin, which is involved in blood clotting. Heparin is usually given by injection, but it can also be given as a nasal spray or a skin patch. Warfarin. Warfarin is a synthetic drug that works by blocking the production of vitamin K-dependent clotting factors. Vitamin K is needed for the production of these clotting factors, so by blocking vitamin K, warfarin can prevent blood clots from forming. Warfarin is taken by mouth. Anticoagulants can be used to treat a variety of conditions, including deep vein thrombosis, DVT. DVT is a blood clot that forms in a vein deep in the body, usually in the leg. DVT can be a serious condition, as it can lead to a pulmonary embolism, PE. Pulmonary embolism, PE. PE is a blood clot that travels to the lungs. PE is a serious condition that can be fatal. Stroke. A stroke is a condition that occurs when blood flow to the brain is blocked. Anticoagulants can be used to prevent strokes in people who are at high risk. Heart attack. A heart attack is a condition that occurs when the blood supply to the heart is blocked. Anticoagulants can be used to prevent heart attacks in people who are at high risk. Anticoagulants can also be used to prevent blood clots during surgery or other procedures. Question 12. Reference values. Reference values are an important tool for interpreting laboratory results. However, it is important to remember that they are just a guide. There is always some variation in the results of laboratory tests, even in healthy people. If you have any concerns about your test results, talk to your doctor. Here are some of the factors that can affect reference values. Age. Reference values can vary depending on age. For example, the reference values for blood pressure are lower in children than in adults. Gender. Reference values can also vary depending on gender. For example, the reference values for hemoglobin are higher in men than in women. Race. Reference values can also vary depending on race. For example, the reference values for serum cholesterol are higher in African Americans than in Caucasian Americans. Medications. Some medications can affect the results of laboratory tests. For example, certain medications can increase or decrease the levels of certain blood cells. Medical conditions. Some medical conditions can also affect the results of laboratory tests. For example, people with diabetes may have higher levels of blood sugar than people who do not have diabetes. Question 13. Plasma proteins. Answer: plasma proteins are proteins found in the blood plasma. They make up about 7% of the total volume of blood plasma. Plasma proteins are essential for many bodily functions, including transporting nutrients and oxygen. Plasma proteins transport nutrients and oxygen throughout the body. Maintaining blood pressure. Plasma proteins help to maintain blood pressure by keeping the blood volume constant. Fighting infection. Plasma proteins help to fight infection by providing antibodies and other immune factors. Clotting blood. Plasma proteins help to clot blood by providing clotting factors. Maintaining fluid balance. Plasma proteins help to maintain fluid balance by preventing fluid from leaking out of the blood vessels. There are three main types of plasma proteins. Albumin. Albumin is the most abundant plasma protein. It helps to maintain blood pressure and transport nutrients and oxygen. Globulins. Globulins are a group of proteins that play a variety of roles, including fighting infection, clotting blood, and maintaining fluid balance. Fibrinogen. Fibrinogen is a protein that helps to clot blood. The levels of plasma proteins can be affected by a variety of factors, including diet. A diet that is low in protein can lead to low levels of plasma proteins. Disease. Certain diseases, such as liver disease and kidney disease, can lead to low levels of plasma proteins. Medications. Some medications, such as steroids, can lead to low levels of plasma proteins. Low levels of plasma proteins can lead to a variety of problems, including edema. Edema is the accumulation of fluid in the tissues. Weakness. Low levels of plasma proteins can lead to weakness and fatigue. Infection. Low levels of plasma proteins can make it more difficult to fight infection. Bleeding. Low levels of plasma proteins can make it more difficult to clot blood. High levels of plasma proteins can also lead to problems, such as kidney damage. High levels of plasma proteins can damage the kidneys. Blood clots. High levels of plasma proteins can increase the risk of blood clots. Question 14. Uses of incubators. Answer: incubators are devices that provide a controlled environment for the growth of microorganisms, cell cultures, or other biological specimens. They are used in a variety of settings, including laboratories, hospitals, and pharmaceutical companies. The common uses of incubators. Growing microorganisms. Incubators are used to grow bacteria, fungi, and other microorganisms. The temperature and humidity of the incubator can be adjusted to optimize the growth of the microorganisms. Culturing cells. Incubators are used to culture cells, such as cells from plants, animals, or humans. The temperature and humidity of the incubator can be adjusted to optimize the growth of the cells. Harvesting cells. Incubators can be used to harvest cells from cultures. The temperature and humidity of the incubator can be adjusted to prevent the cells from dying. Cryopreservation. Incubators can be used to cryopreserve cells, tissues, and other biological specimens. Cryopreservation is the process of freezing biological specimens to very low temperatures, such as -80 degrees Celsius or -196 degrees Celsius. Maintaining temperature-sensitive equipment. Incubators can be used to maintain the temperature of temperature-sensitive equipment, such as microscopes and centrifuges. Sterilizing equipment. Incubators can be used to sterilize equipment, such as petri dishes and pipettes. Question 15. Liver enzymes of diagnostic importance. Liver enzymes of diagnostic importance are alanine transaminase. ALT is an enzyme that is found in the liver and other tissues. It is released into the bloodstream when the liver is damaged. ALT levels are typically elevated in people with liver disease, such as hepatitis and cirrhosis. Aspartate transaminase, AST is another enzyme that is found in the liver and other tissues. It is released into the bloodstream when the liver is damaged. AST levels are typically higher than ALT levels in people with liver disease. Gamma-glutamyl-transferase, GGT is an enzyme that is found in the liver and bile ducts. It is released into the bloodstream when the liver or bile ducts are damaged. GGT levels are typically elevated in people with liver disease, such as alcoholic liver disease and primary biliary cirrhosis. Bilirubin. Bilirubin is a yellow pigment that is produced by the breakdown of red blood cells. It is removed from the bloodstream by the liver and excreted in the bile. High levels of bilirubin in the blood can be a sign of liver disease, such as hepatitis and cirrhosis. Albumin. Albumin is a protein that is made by the liver. It helps to maintain the fluid balance in the body and transport nutrients and hormones. Low levels of albumin in the blood can be a sign of liver disease, such as cirrhosis. Question 16. Water bath. A water bath is a laboratory equipment used to maintain a constant temperature for a liquid. It is a sealed container filled with water that is heated by an electric element. The temperature of the water bath can be adjusted by adjusting the thermostat. Water baths are used in biochemistry labs for a variety of applications, including incubating reactions. Water baths can be used to incubate biochemical reactions at a constant temperature. This is important for many reactions, as the rate of the reaction can be affected by the temperature. Heating solutions. Water baths can be used to heat solutions to a specific temperature. This is useful for dissolving substances or for carrying out reactions that require heat. Sterilizing equipment. Water baths can be used to sterilize equipment, such as pipettes and test tubes. This is done by heating the water bath to a temperature that kills microorganisms. Melting agar. Water baths can be used to melt agar, which is a gelatinous substance used to solidify media for growing microorganisms. Maintaining temperature-sensitive equipment. Water baths can be used to maintain the temperature of temperature-sensitive equipment, such as microscopes and centrifuges. Question 17. Isoenzymes. Isoenzymes are enzymes that catalyze the same chemical reaction but have different physical and chemical properties. They are also known as isozymes or isoforms. Isoenzymes are typically encoded by different genes, but they can also be encoded by the same gene but have different mutations. Isoenzymes are often found in different tissues or organs, and they can also be regulated differently. Isoenzymes are used in medicine to diagnose and monitor diseases. For example, the levels of different creatine kinase isoenzymes can be used to diagnose heart attacks and muscle damage. Here are some examples of isoenzymes. Lactate dehydrogenase, LDH. LDH is an enzyme that catalyzes the conversion of lactate to pyruvate. There are 5 different LDH isoenzymes, and they are found in different tissues. Question 18. Transamination. Answer: transamination is a biochemical reaction that transfers an amino group from one molecule to another. The general reaction is amino acid and alpha keto acid and alpha amino acid. The amino group is transferred from the amino acid to the alpha keto acid, and the alpha keto acid becomes an amino acid. Transamination is catalyzed by enzymes called transaminases. There are many different transaminases, each of which catalyzes the transfer of an amino group from a specific amino acid to a specific alpha keto acid. Transamination is an important reaction in the metabolism of amino acids. It is involved in the synthesis of new amino acids, the breakdown of amino acids for energy, and the production of urea. Transamination also plays a role in the synthesis of other molecules, such as neurotransmitters and proteins. Here are some of the functions of transamination. Synthesis of new amino acids. Transamination is used to synthesize new amino acids from alpha keto acids. Breakdown of amino acids for energy. Transamination is used to break down amino acids for energy. The amino group is transferred to an alpha keto acid, and the alpha keto acid is then oxidized to produce energy. Production of urea. Transamination is involved in the production of urea. Urea is a waste product of protein metabolism. Synthesis of neurotransmitters. Transamination is involved in the synthesis of some neurotransmitters, such as glutamate and GABA. Synthesis of proteins. Transamination is involved in the synthesis of proteins. Proteins are made up of amino acids. Question 19. Internal quality control. Internal quality control, IQC, is a system of checks and balances used to ensure the accuracy and precision of laboratory results. It is an important part of any laboratory, but it is especially important in biochemistry labs, where the results of tests can have a significant impact on patient care. IQC is typically carried out by running control samples alongside patient samples. Control samples are known to have a certain value, and the results of the control samples are used to assess the accuracy and precision of the laboratory's methods. There are many different types of IQC tests that can be used in biochemistry labs. Some common IQC tests include linearity tests. These tests are used to assess the ability of the laboratory's methods to produce accurate results over a range of concentrations. Precision tests. These tests are used to assess the reproducibility of the laboratory's methods. Accuracy tests. These tests are used to assess the overall accuracy of the laboratory's methods. Robustness tests. These tests are used to assess the ability of the laboratory's methods to produce accurate results in the presence of interfering substances. Question 20. Structure of DNA. DNA is a molecule that contains the genetic instructions for all living organisms. It is made up of two long strands of nucleotides that coil around each other to form a double helix. The nucleotides are made up of three parts: a sugar molecule, deoxyribose, a phosphate group, and a nitrogenous base. There are four types of nitrogenous bases in DNA: adenine, A, thymine, T, cytosine, C, and guanine, G. The two strands of DNA are complementary to each other, meaning that the bases on one strand always pair up with the bases on the other strand in a specific way. A always pairs with T, and C always pairs with G. This complementary base pairing is what holds the two strands of DNA together. The double helix structure of DNA is important for several reasons. First, it allows the DNA to be compactly packaged in the cell nucleus. Second, it allows the DNA to be copied accurately during cell division. Third, it allows the DNA to be read and interpreted by the cell's protein-making machinery. Question 21. Normal constituents of urine. Answer: urea, 9.3 grams per dl. Creatinine, 0.670 grams per liter. Sodium, 1.17 grams per liter. Potassium, 0.750 grams per liter. Chloride, 1.87 grams per liter. Question 22. Cap color code of blood collection tube for estimation of. Answer: A. Glucose. A gray vacutainer tube. B. Glycated hemoglobin and lavender. C. Bilirubin. Green. 23. Define pH. Mention the uses of pH meter. Answer: pH is a measure of the acidity or basicity of a solution. It is a scale that ranges from 0 to 14, with 7 being neutral. A pH below 7 is acidic, while a pH above 7 is basic. The pH scale is logarithmic, which means that a change of 1 pH unit represents a tenfold change in the concentration of hydrogen ions. For example, a solution with a pH of 6 has 10 times more hydrogen ions than a solution with a pH of 7. Some of the uses of a pH meter. Agriculture, to measure the pH of soil, which can affect the growth of plants. Aquaculture, to measure the pH of water in fish tanks and ponds, which can affect the health of the fish. Biology, to measure the pH of blood, urine, and other bodily fluids, which can be used to diagnose medical conditions. Chemistry, to measure the pH of solutions, which can be used to control chemical reactions. Food industry, to measure the pH of food and beverages, which can affect their taste and shelf life. Manufacturing, to measure the pH of industrial processes, such as water treatment and wastewater disposal. Pharmaceuticals, to measure the pH of drugs and cosmetics, which can affect their effectiveness and safety. Water treatment, to measure the pH of water, which can affect its potability and suitability for industrial use. Question 24. List 3 electrolytes with normal serum values. Answer: 3 electrolytes with normal serum values. Sodium, 136 to 146 millimoles per liter, mol/L. Potassium, 3.5 to 5.1 mol/L. Chloride, 97 to 105 mol/L. These are just a few of the many electrolytes that are found in the blood. Other important electrolytes include calcium, magnesium, bicarbonate, and phosphate. The normal values for these electrolytes can vary depending on the age, sex, and health status of the individual. Question 25. Mention 3 functions of vitamin A. Answer: 3 functions of vitamin A. Vision. Vitamin A is essential for good vision, especially in low light. It is needed for the production of rhodopsin, a protein in the retina that helps us see in dim light. Immunity. Vitamin A is important for a healthy immune system. It helps the body fight off infections by supporting the production of white blood cells. Growth and development. Vitamin A is important for growth and development. It is needed for the development of the retina, bones, and skin. Question 26. Different types of pipettes. Answer: graduated pipette. This is the simplest type of pipette. It has a graduated scale on the barrel, which allows you to measure the volume of liquid that you are transferring. Graduated pipettes are typically made of glass or plastic. Serological pipette. This is a type of graduated pipette that is specifically designed for transferring small volumes of liquid. Serological pipettes have a long, thin tip that helps to minimize evaporation. Volumetric pipette. This is the most accurate type of pipette. It is designed to deliver a precise volume of liquid, regardless of the viscosity of the liquid. Volumetric pipettes are typically made of glass and are calibrated to deliver a specific volume, such as 100 microliters or 1 milliliter. Air displacement pipette. This type of pipette uses air pressure to draw liquid into the pipette. Air displacement pipettes are available in both manual and electronic versions. Repeater pipette. This type of pipette is designed to dispense the same volume of liquid multiple times. Repeater pipettes are typically used for serial dilutions. Multi-channel pipette. This type of pipette is designed to dispense multiple volumes of liquid simultaneously. Multi-channel pipettes are typically used for assays that require the simultaneous transfer of multiple samples. Question 27. Functions of calcium. Answer: calcium is an essential mineral for good health. A deficiency in calcium can lead to a number of health problems, including osteoporosis, muscle cramps, and high blood pressure. The recommended daily intake of calcium for adults is 1,000 mg for women and 1,200 mg for men. Functions of calcium. Building and maintaining strong bones and teeth. Calcium is the most abundant mineral in the body and makes up about 1.5% of the body weight. About 99% of the body's calcium is stored in the bones and teeth, where it gives them strength and structure. Transmitting nerve impulses. Calcium is essential for transmitting nerve impulses. It helps to carry electrical signals between nerve cells, which allows the body to move and function properly. Contracting muscles. Calcium is also important for contracting muscles. It helps to bind to proteins in the muscle cells, which causes them to contract. Regulating blood clotting. Calcium is involved in the blood clotting process. It helps to activate proteins that form a clot to stop bleeding. Regulating blood pressure. Calcium helps to keep blood pressure at a healthy level. It does this by relaxing blood vessels and making it easier for blood to flow through them. Supporting the immune system. Calcium helps to support the immune system. It helps to produce white blood cells, which fight off infection. Question 28. Scurvy. Answer: scurvy is a disease caused by a deficiency of vitamin C, ascorbic acid. Vitamin C is essential for the production of collagen, a protein that is needed for the formation of connective tissues, such as bones, teeth, and blood vessels. Symptoms of scurvy include fatigue, weakness, joint pain, bleeding gums, and skin sores. Scurvy is easily preventable by eating a diet that includes plenty of fruits and vegetables. Treatment for scurvy is simple and involves taking vitamin C supplements or eating foods that are rich in vitamin C. Question 29. Essential amino acids. Answer: essential amino acids are amino acids that cannot be synthesized by the body and must be obtained from the diet. There are 9 essential amino acids for humans: histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Essential amino acids are needed for the synthesis of proteins, which are essential for many bodily functions, such as growth, repair, and immunity. A deficiency in essential amino acids can lead to a variety of health problems, such as muscle wasting, fatigue, and impaired immunity. The best way to get enough essential amino acids is to eat a balanced diet that includes a variety of protein-rich foods. Good sources of protein-rich foods include meat, fish, poultry, eggs, dairy products, beans, lentils, and nuts. Question 30. Normal values of urea, creatinine and uric acid in blood. Answer: urea, 2.5 to 7.5 mg per deciliter, mg/dl. Creatinine, 0.6 to 1.2 mg/dl for men and 0.5 to 1.0 mg/dl for women. Uric acid, 3.4 to 7.0 mg/dl for men and 2.4 to 6.0 mg/dl for women.